Rat feces are widely suspected to be a source of bacteria that can cause disease in people and domestic animals, such as E. coli and Salmonella. This is part of the reason that we don't like to have rats in our homes, around our food, or on our farms. However, few studies have actually tested rat feces to determine if they could be dangerous. Our goal was to determine if rats from an urban neighborhood and a rural chicken farm carry E. coli and Salmonella. However, not all E. coli and Salmonella are created equal. Some are resistant to antibiotics, while others carry specific genetic codes called virulence genes that allow them to more easily invade the body and cause disease. We therefore also wanted to know if rats carry these more harmful types of E. coli and Salmonella, because they pose a greater health threat compared to other strains. What we found was that many rats were completely free of E. coli and Salmonella, while others carried strains that weren't particularly harmful. However, a subset of the rat population carried strains of E. coli and Salmonella that were resistant to antibiotics, had virulence genes, or both. For example, over 25% of rural rat E. coli was resistant to multiple antibiotics and had virulence genes. What was most interesting, however, was that the characteristics of E. coli and Salmonella in the rural rats was significantly different from what we found in the urban rats. E. coli and Salmonella in the urban rats was most similar to what would be found in an urban environment. For example, some urban rats carried strains of E. coli that were highly resistant to antibiotics and would normally be found in sewage. Rural rats, on the other hand, were carrying types of E. coli and Salmonella more commonly associated with an agricultural environment. We concluded that the presence of harmful E. coli and Salmonella is not a characteristic of rats themselves. Rather, it's a result of the bacteria that are present in their environment. Rats appear to act like a sponge that can soak up the bacteria they come in contact with. We also know that when bacteria get together, they can trade segments of DNA that allow them to cause disease or to be resistant to antibiotics. Therefore, the ability of rats to carry a variety of different bacteria from different sources could facilitate this sort of gene transfer and result in the development of bacteria that are more dangerous than what were present in the first place. Although further study is needed to determine whether the bacteria in rat feces can or is being transmitted to people and domestic animals, our results suggest that rat feces can indeed be dangerous, but that the dangers associated with rat feces are more complicated and nuanced than previously appreciated.